Good day wishes to dear students. I welcome you to the course Enzymology. Under the course Enzymology, we have Module 1, the basics of enzymes. I have divided the Module 1 into two parts. That is the part 1, Introduction to Enzymes, and part 2 is about the strategies involved in purification of the enzymes. So when we try to understand what the enzymes are biocatalysts. They are large protein molecules and are only required in very low concentrations. So what do these enzymes do during the reaction? They speed up the reactions, but the thing is they are not consumed during the reaction. So that is what we have to remember. They accelerate the rate of the reaction without themselves being consumed during the reaction. We have a beautiful illustration over here. We have the substrate molecules. We have an enzyme molecule. There is something called as active site. So the substrate molecules bind to the enzyme, forming a complex called as enzyme substrate complex. After the reaction, the product is formed, releasing the free form of the enzyme. So enzymes, they are not consumed during the reaction, but they speed up the reactions. So we know that enzymes are protein molecules, but many enzymes, they require a non-protein component for the activity, such as cofactor, coenzyme, and prosthetic group. Now the cofactors, they are small inorganic ions, mostly metal ions, such as copper in cytochrome oxidase, magnesium in kinases, and iron in catalase and peroxidase. Then we have coenzymes. They are small non-protein but organic compounds, such as coenzyme A, vitamin B1, B2, B6, B12. So the difference between a coenzyme and a cofactor is cofactors, they are inorganic ions, and coenzymes, they are organic compounds. Then we have prosthetic group. They are tightly bound large complex organic molecules such as heme. So what happens in enzyme substrate complex formation? How do the complex forms? The part of the enzyme combining with the substrate is the active site. So active site has certain features. The characteristic features of the active sites are, they are the pockets or clefts in the surface of the enzyme. So the R groups at active site, they are referred to as catalytic group, and the shape of the active site is complementary to the shape of the substrate. The enzyme attracts and holds the substrate using a non-covalent weak interactions, and the conformation of the active site determines the specificity of the enzyme. So these are some of the important characteristic features of the active site. That is, they are the pockets in the surface of the enzyme, and the shape of the active site is complementary to the shape of the substrate, and there is a weak non-covalent interaction between the enzyme and the substrate, and conformation of the active site determines the specificity of the enzyme. So as I said earlier, they are the portion of the enzyme which folds precisely so that the substrate molecule fits in, or the contours of a substrate fits precisely to the active site of the enzyme through electrostatic interactions, which facilitates the bond reactivity. Then we have something called as allosteric site. So this is a site other than the active site. So we will be discussing about allosteric site in allosteric regulation. Moving on further, there is a beautiful model to explain the enzyme reactions or the enzyme substrate reaction. So this model is divided into three phases. The phase 1 is about the initiation, the phase 2 is about the transition state facilitation, and the phase 3 is about the termination. So what happens in initiation? Let us carefully understand. Here the reactants or the substrates, what we are referring to, bind to the active site in a specific orientation. So this is very, very critical. So binding to the active site is not only the criteria, binding to the active site in a specific orientation should also be considered. Only then the enzyme substrate complex is formed. So reactants bind to the active site in a specific orientation, forming an enzyme substrate complex. If there is no specific orientation binding, then you cannot expect the enzyme substrate complex to be formed. Then in the second phase, we have transition state facilitation. What happens here? Here, the interaction between the enzyme and substrate, it lowers the activation energy. We will see what the activation energy in the next few slides. So the interaction between the substrate and the enzyme is lowering the activation energy. That means the energy required for the reaction to happen is at very low level. So interaction between the enzyme and the substrate lowers the activation energy required. Now what is happening in the third phase, that is the termination phase? Products have lower affinity for active site. So in the initiation phase, the reactants or the substrates has higher affinity for the active site. In the termination phase, products have lower affinity for the active site. So since they have lower affinity for the active site, there is no binding to occur. So the products are released, and also enzyme is unchanged and released in the free form. So they are not being used up during the or consumed during the biochemical reactions. So the model of enzyme action is beautifully explained, taking into consideration the three different phases, that is initiation, transition state facilitation, and termination. So as I said earlier in the previous slide, the minimum quantity of energy which the reacting species must possess in order to undergo a specified reaction is referred to as activation energy. So it is worth to understand the activation energy using this beautiful illustration. So this represents the reaction without enzymes, and this represents the reaction with enzymes. So if you just look at this bar which denotes the energy level, without the enzyme the reaction requires a lot of energy. So in the presence of the enzyme, the energy required is very less. So the interaction between the substrate and the enzyme lowers the energy of activation. So activation energy is lowered, that means they require less energy, so for the reaction to happen, and immediately the product is formed. Those speed up the reaction too. To understand why reactions have an activation energy, the first and foremost is two molecules have to collide, so thereby organizing the system. So not only do they have to be brought together, they have to be held in exactly the right orientation. See, the thing is, when two molecules have to collide, they have to be in close proximity, but it doesn't mean that that is sufficient. They have to be held in exactly the right orientation relative to each other to ensure that the reaction can occur. So both these factors raises the free energy. Now what is free energy? It is a capacity of a system to do work. So when these factors raise the free energy of the system by lowering the entropy. So entropy is lowered, so free energy is increased. So thus the interaction between the enzyme and substrate lowers the activation energy. When we say this, only a small fraction of the collision occurs between the substrate molecules, converting the substrate into the products of the reaction. So a small fraction of the collision occurs between the reactant molecules, converting the reactants into the products of the reaction. That is how the interaction between the substrate and the enzymes lower the activation energy. So like all proteins, each enzyme molecule has a particular shape, and this particular shape determines which chemical reaction the enzyme can speed up. We have two beautiful models to explain the enzyme substrate complex formation. The number one is the lock and key model. Here the enzyme molecules bind to the complementary shape of the substrate. So exactly the complementary shape of the substrate is bound to the enzyme molecule. And we have another model to explain the enzyme substrate interaction, that is the induced fit model. Here what happens, there need not be exact complementarity before binding. So what happens here is the substrate binds to the enzyme, changing the conformation of the enzyme so that the substrate fits to the active site region of the enzyme. So it induces a conformational change, allowing the substrate molecule to bind to the enzyme. So you can see here enzyme shape changes slightly, allowing the substrate to bind to the enzyme, forming enzyme substrate complex, and finally the enzyme and products are released independently and enzyme is not being used up in the reaction. So these two models are beautifully explained about the enzyme substrate complex formation. Now, do the enzyme activity is altered or affected by various variables, that is key variables. Here the key variables are the temperature, the pH and the concentration. Temperature, pH and concentration of the enzyme or molecules do vary the rate at which the enzyme functions. So what happens if there is any change in the temperature? So we have the enzyme and we have the substrate. There is a complementarity between the enzyme active site and the substrate molecules. So if there is a slight change in the temperature that is above the optimum temperature of that particular enzyme reaction, say for example above 40 degrees in this case, the enzymes are denatured. So when the enzymes are denatured, that means the active site of the enzyme changes the shape. You can see or observe over here, this was the shape of the enzyme prior to heating, and now the enzyme has changed the shape. The active site of the enzyme changes the shape. So that means we are saying it as the enzymes are denatured. So the active site of the enzyme changes the shape and can no longer bind to the substrate molecule. Same thing happens if there is a change in the pH of the reaction mixture. So we have the enzyme, the active site of the enzyme, and there is a complementarity shape for the substrate to bind. If there is change in the pH of a solution that is maybe very acidic or very alkaline, the complementarity between the enzyme substrate is not observed because the enzyme active site changes the shape, the enzyme undergoes denaturation, so the substrate molecules cannot bind to the active site of the enzyme. So in both the cases, the same thing happens, and it is also implied that the concentration of the enzyme or the concentration of the substrate also affects the enzyme substrate reactions. So moving on further, the enzymes are highly specific to their substrate and reaction being catalyzed. This is the specificity of the enzymes. Say for example, the enzymes show bond specificity. That is bond specificity of the enzymes. Here you can see the specificity is towards the peptide bond, and here also it is about the peptide bond. And then similar to bond specificity, there is something called as stereospecificity. You can observe over here, it is the L-alanine oxidase and L-alanine, and this is L-alanine oxidase and D-alanine. So there is the stereospecificity. See, you cannot have the L-alanine oxidase and D-alanine because the enzymes are stereospecific. It requires only the alanine in the L form, so the reaction doesn't happen over here. Only when there is L-alanine, the reaction happens. So enzymes, they are highly specific to their substrate and the reaction being catalyzed. Now, are the enzymes saturable? Yes, this means as more substrate is added, the reaction rate will increase because more active sites becomes occupied. This can continue until the enzyme becomes saturated. So when we say saturated, that means all the active site portions are completely filled up by the substrate molecules. So this can continue until all the enzyme becomes saturated with the substrate and the rate reaches a maximum. So we say that as the reaction is proceeding in a maximum velocity. Now moving to the next issue, that is nomenclature and classification of the enzymes. So enzymes are often classified by placing them in categories according to the reactions they catalyze. We have oxidoreductase. The important subclasses of this class is the dehydrogenases, oxidases, peroxidases, reductases, monooxygenases, dioxygenases. Then we have transferases. The important subclasses are the C1 transferases, glycosyl transferases, aminotransferases, then phosphotransferases. We have hydrolases. The subclasses are esterases, glycosidases, peptidases, amidases. Then we have lyases. The subclasses are the CC lyases, CO, CN, CS lyases. Then we have isomerases, The subclasses are epimerases, cis-trans isomerases, intramolecular transferases. Then we have ligases, that is CC ligases, CO, CN, CS ligases. So to put together, there are 6 classes of enzymes based on the reactions they catalyze, and there are several important subclasses of the enzymes. So the Enzyme Commission in 1955, that is International Union of Biochemistry and Molecular Biology, gave the 4-digit numbering 1, 2, 3, 4. That is first digit, that is it represents one of the 6 major classes of the enzyme activity, and the second digit represents the subclass, that is the type of the substrate or bond cleaved, and the third digit represents the subclass, that is group acted upon, the cofactor required, etc. And the fourth digit represents a serial number, that is order in which the enzyme was added to the list. So the 4-digit numbering system, which refers to as 1, 2, 3 and 4, is the first one. It is about the one of the 6 major classes of the enzyme activity. The second one is about the subclass, that is the substrate or the bond cleaved, and the third one is the subclass, that is group acted upon, the cofactor required, etc. And the fourth one is a serial number, that is order in which the enzyme was added to the list. Next, how to denote the activity of the enzyme. So units for expressing the enzyme activity. Number 1 is the reaction rate. So this implies the substrate utilized per unit time or the product form per unit time. Then enzyme activity, it is defined as the amount of enzyme converting 1 micromole of substrate per second. Then what is the turnover number? It is another common term, that is the number of substrate molecules converted by one enzyme molecule under specified conditions. And the specific activity refers to enzyme activity per mg of protein or per mass of the protein. That is, all the protein in a sample may not be the enzyme. So that is the reason why it is taken as per mg or per mass of the protein. So this unit also gives us an indication of the purity of the enzyme. That is, an impure enzyme will give low activity per unit mass. So how to define the enzyme activity? More specifically, 1 unit activity, that is equal to 1 international unit. The amount of the enzyme which converts 1 micromole substrate per minute at 25 degrees centigrade. And 1 unit of specific activity is defined as the enzyme activity per milligram of total protein present. That is, for example, 10 micromole per minute per milligram of protein, or 10 international units per milligram of protein. So this is about determining the activity or units of determining the enzyme activity. So as I said earlier, the part 2 of the module 1, that is purification of the enzymes. So we'll try to understand what are the strategies for purification of the enzymes. To understand the behavior of an enzyme in a very complex system, it should be understood in a simple system. So that means we need to isolate the enzyme from a very complex system. So we are saying it is a complex system, that means it is truly a very much complex system. So it is very difficult to isolate the enzyme in a very pure form. So let us try to understand the basic strategies in isolation and purification, and as well as characterization of the enzyme. So any aim of a purification process is to maximize the yield. That is, the maximum possible yield should be taken care of. That means the activity should be retained in the recovered product. The maximum catalytic activity should be there. So one thing is yield. You should have maximum yield. Then whatever the yield you have should have maximum catalytic activity. And when you fulfill these two, the third important criteria is maximum possible purity. So no other enzymes or large molecules should be there or present as impurity, because purification of an enzyme is very critical. So you should have more yield, you should retain the catalytic activity even after purification, and possibly the purity. So how to determine all these things? You should have a suitable assay procedure required to measure the yield, required to measure the activity, required to measure the purity level also. So a suitable assay procedure is required. So units, of course, it is micromole per minute, and specific activity is a microgram of protein. So this is a flow chart which gives us an overview of the strategy in enzyme purification. Here we have the source of the enzyme, could be animal, microbial or plant. Then if you go for partial homogenization, that is subcellular fractionation, you will get the purified organelles, and then you can go for extraction by extraction or dispersion method. Then you have the extract, and then use the extract to obtain the pure form of the enzyme by using separate separation methods, that is chromatographic separation methods, more specifically the affinity chromatography. And if it is not possible, you can directly homogenize, that is total homogenization of the source, so you get the extract. If you are not interested with some of the enzymes from the organelles, so you can directly go for total homogenization of the source, then you get the extract. Then you can go for separation methods, that is large scale separation methods, so you will get a crude extract, that is crude preparation. Then you can go for small scale separation methods to obtain the purest form of the enzyme. If you ask me, does this work? Yeah, it is the basic outline or basic design or strategy in enzyme purification, and each of the step requires a lot of understanding or studies and trial and errors to come to this phase, that is purest form of the enzyme. Of course, this is the fundamental or the basic strategy to obtain the purest form of the enzyme. So we are speaking about the source. What is the source? What should be the choice? How should be the choice of the source? A source should be in which the required enzyme occurs in large amounts. So if you select a source where the enzyme amount is very less, then probably you will be not having sufficient concentration of the enzyme or the quantity of the enzyme for further analysis. So you should make sure that the source what you have chosen is having the required enzyme concentration, that is in large quantity, and a suitable expression system for the recombinant enzyme. If it is a recombinant enzyme, there should be a suitable expression system. So it can be prokaryotes, eukaryotes, yeast, insect cells. So all these things make up the source, but it depends upon your experimental aim which source should be selected. But when you are selecting, you should keep these two things in your mind. If it is a recombinant enzyme, suitable expression system should be available, and the enzymes in which the source you are trying to isolate should be in large amounts. Now, if you just go for methods of homogenization, if it is a mammalian tissue, it lacks a rigid cell wall, so it is easy homogenization process. If it is plant, fungal or bacterial, they have a rigid cell wall, so it is a little harsher methods are required. So grinding with abrasives, you can go for freeze and thaw, then longer periods of blending, addition of glass beads and etc. And extraction of membrane bound enzymes, you can go for some of the detergents, so that the membrane bound enzymes are easily isolated. So these are some of the methods of homogenization specific for the source which lacks rigid cell wall as well as which has a rigid cell wall. Then you can see over here by using several properties such as size or mass, you have centrifugation, gel filtration, dialysis. Then based on polarity, you have ion exchange chromatography, then chromatofocusing. Then based on hydrophobic character, you have hydrophobic chromatography. Then specific binding, based on specific binding sites, you have affinity chromatography, affinity elution. So these are some of the important methods of separation based on size, charge, solubility and the position of specific binding sites. So choice of the separation methods, that depends on the scale of the preparation and yield of the enzyme required, the time available for the preparation, that is very important, and the equipment and expertise that is available in your lab. So earlier stages, that is large scale, the methods based on changes in solubility, and later stages, that is small scale preparations, you can go for column chromatography or electrophoresis. So in large scale separation methods, you can only observe the changes in solubility. So in later stages, that is in small scale separations, you can go for column chromatography or electrophoresis. So you have purified the protein. So now how to judge the success of purification procedure? Purification procedures, or the success of a purification procedure, is given by the purification table. So it is measured for each purification step. So these are some of the important points to be remembered when we go for purification table analysis. Number one is the volume of the enzyme solution. The number two is the protein content of the solution. Number three, the activity of the enzyme solution, which is expressed in units per ml. The protein content is expressed in milligram per ml, and volume of solution is expressed in ml. So as I said earlier, the total amount of enzyme, that is denoted as U, is given by activity multiplied by volume of the enzyme. That is units per ml multiplied by volume, that is ml. Specific activity is given by activity divided by protein content. Now how to measure the percentage of yield, that is total amount of enzyme after a purification step divided by total amount of enzyme before that step. That step means over here it is the previous step that is adopted in the purification. Say for example, if we are going for chromatographic method, affinity chromatography, and that if that is supposed to be the third method, and in the second method if we have adopted gel filtration chromatography, so here the total amount of enzyme after purification step, that is after gel filtration, that divided by total of amount of enzyme before that step. So before gel filtration, what the step you have adopted, what was the total amount of enzyme. So that is how the yield is calculated. Now how to calculate the purification factor? Specific activity of enzyme after a purification step divided by specific activity before that step. So if you just look at the purification table, it's a very generic table. So yield keeps on decreasing and the purification factor increases. So the yield keeps on decreasing because you are removing the unwanted compounds or molecules, and there is increase in the specific activity as you adopt several separation methods, and purification factor is also increasing. So how to calculate the purification factor or the yield? So I have given it in the previous slide. So yield is calculated by taking the total amount of enzyme after a purification step divided by total amount of enzyme before that step, and the purification factor is calculated by taking specific activity of enzyme after a purification step divided by specific activity before that step. Okay, now how to characterize the enzyme? Some of the important physical properties to characterize the enzymes are the native molecular weight determination. So to determine the native molecular weight, then subunit molecular weight determination, indicating whether the enzyme protein is monomeric or oligomeric. If oligomeric, whether as identical subunits or different types of subunits. So this is how you characterize the enzyme. And some of the other properties are identifying the number of sulfhydryl group determination. Then absorption spectrum, indicating whether it is a simple protein or has a cofactor bound. End group determination. Chemical properties like optimum pH, optimum temperature, energy of activation, and kinetic properties. So these are some of the important characteristics of an enzyme that can be determined. So a suitable assay method has to be developed. So to determine the purity, to determine the yield, to determine the activity. So enzyme assay methods are very, very important. The assay is the act of measuring how fast a given amount of enzyme will convert the substrate to product. That is act of measuring a velocity. So an assay requires to determine the concentration of a product or substrate at a given time after the starting of the reaction. So different enzymes require different estimation methods depending on the type of the reaction they catalyze, the nature of substrate or product or coenzyme. Okay, so we have fluorescence method, we have spectrophotometric, we have sampling, we have electromanometric and polarimetric methods. So I'll take some one or two examples. Most commonly used is spectrometric photometric methods. Many substrates and products of enzyme reactions, they absorb light either in the visible region or in the UV region. So the conversion of one into another is followed by considerable change of absorption, and by measuring this change, the progress of the reaction can be followed quantitatively. So mostly the spectra of substrate and the product are not the same. So they are different for each. The enzyme is allowed to react with the substrate, and the decrease in the concentration of the substrate or increase in the product produced will be followed spectrophotometrically. So what are the advantages of this method? The advantages is the method is easy, it is simple, it is sensitive, require small sample concentration, and the entire process can be followed quantitatively. Right? So we have a coupled enzyme assay. When the enzyme reaction being studied cannot be made to give an absorption change. So what to do? The spectrophotometric method will be used by adding another purified enzyme which acts on the product of the first reaction to cause a change. So you are using the two different enzymes. So first enzyme cannot measure any absorption change, either the substrate or the product. So what you do is you add a second enzyme which acts on the product of the first reaction, and it gives the second product, but in between there is change in the absorption, so you can follow it up. So this helps to follow the first enzyme reaction. You can see over here there is aconitase and there is isocitrate dehydrogenase. So NAD plus has no absorption at 340 nanometers, but NADH has absorption at 340 nanometers. So you can measure the reaction of the first phase. So this is called as coupled enzyme assay. So the first enzyme must be rate limiting enzyme. It is often difficult to match ideal conditions for two distinct enzymes. So these are some of the limitations using coupled enzyme assays. Inhibitors or substrates may affect both the enzymes. So that is an important caveat for the coupled enzyme assays. Then we have fluorescence method. Fluorescence is when a molecule emits light of one wavelength after absorbing light of a different wavelength. Uses a fluorometer and flavin compounds which fluoresce strongly in the oxidized form and lose their fluorescence in the reduction phase. So this can be used for following enzymatic oxidation reduction reactions. What are the advantages of this method? It is highly sensitive, that is much more sensitive than the spectrophotometric assay. Detection in small quantities is possible, and it is safe, that is it is not dangerous to the person working, and it is sensitive to the environmental conditions. So these are some of the important fundamental concepts I wanted to discuss with you. So thank you very much for your interest and patience listening. Thank you.